Dear student, welcome to Biospirit. In last class, we discussed living world to morphology of flowering plant, fixed questions and answer. In this video, we are discussing chapter 6 to 12, the fixed questions and answers. So, chapter 6 is anatomy of flowering plant. So, from anatomy of flowering plant, first questions arise like that: what are buliform cells? Write its significance. Buliform cell definition is there: upper epidermal cells and veins modified to form a large empty colorless cells is called buliform cells, and its function is that it is folding or curling of the leaves and it remove the excess of water. And next question is that what is stomatal aperture? Explain the structure of stomata with label diagram. So, what is stomatal aperture means? A group of cells that control the exchange of gases and water in plant. So here two types are there. So one dicot type of stomata, another one monocot stomata. So in dicot they have a kidney shaped stomata, and in monocot they have a dumbbell shaped stomata. So both type I drawn because both are important diagrams are there. Next question: what is a vascular bundle? Name any two type of vascular bundle. So vascular bundle definition: the xylem and phloem are present in the together. It is helpful for the water and food transport. The whole bundle we are calling as a vascular bundle. And here two types are there: one radial, another one conjoint. Radial means the xylem and phloems are arranged in circular manner, and conjoint means the xylem and phloems are present in the up and down in parallel. So in conjoint they have two types: one is their open type of open type of vascular bundle, another one closed type of vascular bundle. Open type means between xylem and phloem they have a cambium, means open type. Between xylem and phloem they don't have a cambium, means closed type. Next question is it. So fourth question: differentiate between exarch and endarch xylem with suitable example. So exarch means the protoxylem is towards the periphery, and where metaxylem is towards the center. It is present in root, and metaxylem is towards the periphery, whereas protoxylem is towards the center. It is exact opposite to the exarch is there. So endarch example, that is stem. You have to remember, like eggs means outside, end means inside. Eggs means protoxylem, means small xylem tubes are present in outside. End means large xylems are present at the outside. Next question: what is trichome? Write its significance. So trichome means fine outgrowth on the plant that protect from the environmental hazards, means they protecting by animals or some environmental hazards. And the trichomes, it is helpful for the protection purpose, and it prevents the evaporation of water. And next question is write any 6 features of TS of monocot stem. So I written these 6 points, most easiest points are there, and you can refer it. And next question is write any 6 features of TS of dicot stem. And here also I mentioned the outer layer is called epidermis, and just above the epidermis they have some waxy layer is called cuticle. And these are the similar points are there in all stem, leaf and root. You can refer it. And next one is write any 6 features of TS of dicot leaf. You can read. These are the continuing points are there. Next, write any 6 features of TS of monocot leaf. And monocot leaf means you have to remember, so always the buliform cells. Next, write any 6 features of dicot root. From this chapter, last question is write any 6 features of TS of monocot root. So these are the points are there. Here one more thing is there, students. So maybe they will arise the 6 features, otherwise they will ask the diagram. So you have to be ready for the diagram also. So they will ask, draw a neat label diagram of dicot stem, like that. So you have to practice all 6 diagrams. So next chapter is there, chapter number 7th, structural organization in animals. So first question, draw a neat label diagram of alimentary canal of frog. So this diagram is there. Next question, draw a neat label diagram of male reproductive structure. You have to practice this diagram. And next question is there, again, draw a neat label diagram of female reproductive structure. So from these 3 diagram, anyone they will ask for the 5 marks question. So you have to be ready. Next, write any 6 external characteristics of frog. So you know very well external characteristics of frog. And next you have to refer these points. Next chapter is chapter number 8, cell structure and function. So from this chapter, first question, who discovered nucleus? The nucleus is discovered by Robert Brown. List the function of a plasma membrane. It acts as a protection. It helpful for the transportation of oxygen, CO2 and food molecules. It gives the cell shape and support. It allows cell to communicate and signal each other. It acts as a cell recognition, cell potential, endocytosis, and exocytosis. And next question is there, draw a neat label diagram of mitochondria. So it is a diagram, you have to practice it. And next is there, draw a neat label diagram of plant cell. So it is also plant cell diagram is there, you have to practice. Next question is there, 5th one, why are mitochondria called powerhouse of the cell? Because it releasing energy from food. And next question is there, 6th one, ribosomes are a protein factory of the cell. Comment. So ribosomes, it is helpful for the synthesis of protein, so that's why we are calling as a protein factory of the cell. And next, mention any 2 type of the plastid with its function. So total 3 types are there, so I mentioned all 3, but there is only 2, so I have written 1 extra. You have to read all 3. Sometimes they will ask 3 also. So first one, chloroplast. It is responsible for the photosynthesis because it contains chlorophyll pigment. And second one is their chromoplast. Chromoplast, it is helpful for the coloring purpose. Coloring of it gives color for the flower and fruit. And you know very well the green chili they converting into a red chili, and green banana they converting into a red banana. Next question is their 5th one, why are mitochondria called as a powerhouse of the cell? Because it releasing energy from food, so that's why we are calling as a powerhouse of the cell. And second one is a ribosomes are called protein factory of the cell. Comment. Because it is helpful for the protein synthesis, so that's why we are calling as a protein factory of the cell. And next, mention any 2 type of the plastid with its function. So here there I see 2 type of the plastid, but I mentioned 3 types. How to read all 3 because sometimes they will arise 3 types. So first one is there chloroplast. It is responsible for the photosynthesis. Next one is there chromoplast. It gives color for the flower and fruit. And you know very well, green chili they converting into a red chili, and green capsicum it is converting into a red capsicum. Green banana they converting into a yellow banana. So because of that is chromoplast. Why it is converting green into red means because the chloroplast it is converting into chromoplast, so that's why they giving some other color. And next, leucoplast is there. It is colorless and its function is there. These are colorless plastid store the food in plant. So in leucoplast, based on that is what type of food storage is there, they are classified into 3. First one, amyloplast. It is responsible for the starch storage. Second one is their elioplast. It is helpful for the fat storage. And third one is their aleuroplast. It is helpful for the protein storage. And next, what are mesosome? Write its significance. So it is formed by the invagination of the plasma membrane into the cell. So that invagination we are calling as a mesosome. So it is visible in bacteria, that is prokaryotic cell, and its function is their DNA replication. It means duplication of a DNA, and respiration and reproduction, and next one is their secretion. And next question, how are eukaryotic cells differ from prokaryotic cells? So prokaryotic cell don't have any well-defined nucleus. Eukaryotic they have a true nucleus. It means well-developed nucleus. And prokaryotic lack of membrane bond organelles, and eukaryotic they contain membrane bond organelles. And prokaryotic they have a 70S ribosome. Eukaryotic they have a 80S ribosome. And prokaryotic single circular DNA is there, and eukaryotic they have a linear DNA. And prokaryotic they don't have any histone protein, but in eukaryotic they have a histone molecules, histone proteins. And next is mention the functions of cytoskeleton in this cell. It first one is provide structural support, and second one it help in the cellular movement, and third one intracellular transport, and it act as a endocytosis and exocytosis. Next question is what are the difference between plant cell and animal cell? You know very well plant cell and animal cell difference. First one is the plant cell they have a cell wall, animal cell they don't have a cell wall. And plant cell they contain 90 to 92% of vacuole, and in animal cell they have a very small vacuoles and many in number. And plant cell large in size and it is smaller in size. The plant cell have a plastid, but animal cell they don't have a plastid. And centromere are absent. In animal cell they have a centrosomes. Sorry, here I pronounced centromere. So centrosome are absent, centrosome are present. Next, plants they don't have any cilia, and animal cell have a cilia. Lysosomes are very rare in the plant cell. Animal cell they have a lysosomes. Next, draw a neat label diagram of the animal cell. So last year there is the question on maybe animal cell diagram. So they will ask fix animal cell or plant cell one diagram sometimes. And next question is explain the fluid mosaic model of the plasma membrane. So here I mentioned 2 points: who discovered and what is the component of the fluid mosaic model. It is given by Singer and Nicholson, and it is contain phospholipid, cholesterol, protein and carbohydrate. Next, these are the fluid mosaic model. Continue points are there, you can read, and diagram also how to practice. And next question is there, describe the structure of chloroplast with the help of neat label diagram. You have to describe the structure of chloroplast. Next question, what are chromosomes? How are they classified based on the position? So here we have to write definition of chromosome. The chromosomes are thread-like structure found within the nucleus, and the types are there: metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. The with explanation, they have a diagram also, you have to practice. Next question, 17th question, describe the structure of nucleus with the help of neat label diagram. So these are the nucleus points are there, how to read. The last question is there, that is from cell structure and function chapter. So that is write a short note on cilia and flagella. These are the cilia and flagella points are there, you can read it. Next chapter, the chapter number 9th one, biomolecules. So from biomolecules, the first question is there, name the nitrogenous base of DNA molecules. So how to write 4 nitrogenous base of DNA, that is adenine, thymine, cytosine, guanine. And next question is there, what are the polysaccharides? Give one example. So long chain of carbohydrate molecules composed of several small monosaccharides. So it's called polysaccharides. Example, starch, glycogen, and cellulose. Next is there, what are the protein? Name the bond present in the protein. So how to write, there is protein definition and which bond is present. Protein are the building block of amino acid is there. We can write in short form also, that is building block of amino acid. Otherwise you can refer this definition also. And the protein, they have one bond, that is we are calling as a peptide bond. Next question, mention the functions carried out by a protein in living organism. So that is structural support, biochemical catalyst, hormone, enzyme, building blocks, initiators of the cellular death. And next question is there, name the monomer of the insulin. The monomer of the insulin is fructose is there. What is biomacromolecules? Large complex organic compounds that are essential for life in living organism is called biomacromolecules. Next question is there, explain the nature of enzyme action. So these are the points are there, you can read it. Next question, classify the enzyme based on the type of reaction they catalyze. It is answer, you can read it. Next question is describe the secondary structure of DNA. So that is, DNA is a double helix structure. The double helix structure was first discovered by Watson and Crick. In a double helix, the two DNA strands are held together by hydrogen bond between nitrogenous base from forming a base pair. Next point is there, in DNA, adenine pair with thymine, guanine pair with cytosine. Each DNA strands run in an anti-parallel orientation, and one strand run 5' to 3', while other strand run 3' to 5'. Next question is that describe the important properties of enzymes. These are the important properties, you can refer it. Next, name the various chemical component of the DNA and RNA. So that is, DNA is in one column you have to write DNA, another one column RNA. DNA is a deoxyribose sugar, RNA is a ribose sugar. DNA use the base pairs adenine, thymine, cytosine and guanine. In RNA they use adenine, uracil, cytosine, guanine. In place of thymine they use uracil. And next question is there, list any 3 secondary metabolites with example. So alkaloid is there. Next, terpenoids is there. Phenolic compound, latex. And next one is there, write the classification and nomenclature of the enzymes. So it is the answer, you can refer it. And next question is there, classify the carbohydrate with example. That is monosaccharides, oligosaccharides, polysaccharides. Monosaccharides they have a example glucose, fructose, ribose. Oligosaccharides they have a sugar, maltose, lactose. Polysaccharides they have a starch, cellulose, chitin. Next question, write a short note on polysaccharides. There are a long chain of the carbohydrate molecules composed of several small monosaccharides is called polysaccharides. So next is what are protein? Name the most abundant protein present in the plant and animal cell. So first definition is protein definition. The protein are very large molecules composed of a basic unit is called amino acid. So in plant they have, in animals they have abundant protein, that is collagen. In plants they have that is rubisco. Rubisco full form is ribulose, bisphosphate, carboxylase and oxygenase, and it is they arising expand the they arise the question on the 1 marks, that is expand rubisco. Like they will give expand rubisco, they will give 4 options, you have to choose anyone. For that you have to remember that this full form. Next, write a short note on cofactor. So these are the cofactor points are there, you refer it. Next chapter, that is chapter number 10, that is cell cycle and cell division is there. So first question, what are the significance of the mitosis and meiosis? So first we will write significance of the mitosis, that is it help in the growth, it helps in the repair and regeneration, it help in maintaining genetic stability, and it helps in asexual reproduction in lower organism. And in meiosis significance, it helps in haploid gamete formation, it is helpful for the sexual reproduction, it lead for the exchanging of a genetic material, and it is also helpful for the evolution also. Next question, list any three difference between mitosis and meiosis. So in one column you have to write mitosis, in another column you have to write meiosis. So you know very well mitosis is a equational division, meiosis is a reductional division, and mitosis it is a process by which the cell divide into two daughter cell, and meiosis it is a process by which cell divide into two daughter cells. And here main difference is that each having a, in mitosis each having exactly identical number of chromosome, but in meiosis each having half number of chromosomes. And in mitosis it is occur in somatic cell, it is occur in meiosis occur in reproductive cell. And next fifth one is the stages of mitosis is prophase, metaphase, anaphase, telophase. In stages of meiosis they have a two type, that is part 1 meiosis 1, second one that is meiosis 2. In meiosis 1 they have prophase 1, metaphase 1, anaphase 1 and telophase 1. In meiosis 2 they have a prophase 2, metaphase 2, anaphase 2 and telophase 2. And next, explain the phases of cell cycle. So here they have two main phases in cell cycle. One is the interphase, another one M phase. In interphase the Z1 will come and S phase, Z2 phase. In M phase they have prophase, metaphase, anaphase and telophase. So these points you can refer. Next question, explain the three stages of mitosis. That is prophase is there. In prophase cell organelles are disappear, nuclear membrane and nucleolus are disappear, chromatin start condensation. In metaphase the chromosomes are maximally condensed, chromosomes are arranged in equatorial plate, centriole reach at the both the pole region. Next phase is anaphase, the chromosomes are start moving both the pole, centromere starts splitting. Last one is a telophase, nuclear membrane, nucleolus and cell organelles are reappear, chromatin materials decondense, and finally cell divides into two daughter cell. And next question is that explain the stages involved in the prophase 1 of the meiosis. So there are 5 stages are there. First one that is liptotin, second one zygotin, third one pachytin, the fourth one diplotin, fifth one dikinesis. In liptotin, so nucleolus, nuclear membrane, cell organelles are start disappearing and centriole start moving both the pole region, and these common points are there from liptotin to dikinesis, and only one few difference are there. In liptotin the chromatin start condensing. In zygotin the between two chromosomes, that is synaptonemal complex is develops. Next pachytin, so in pachytin the recombination is takes place between the two homologous chromosome and they become as a egg-shaped structure, that is we are calling as a chiasmata. And next diplotin is there. So I mentioned point, you can refer from this question. Last point is there, that is dikinesis, that is also I mentioned here, you can read it. And next, that is define the following terms. First question is there, define cell cycle. It is a process by which cell grows and divide to form as a two daughter cells, is called as a cell cycle. And next question is there, what is karyokinesis? Karyokinesis means nucleus division. Next, what is cytokinesis? Cytokinesis means division of cytoplasma. Synapses means the homologous chromosomes start crossing over, and that is we are calling as a synapses or pairing of the homologous chromosome. Next chapter is there, that is chapter number 11th, photosynthesis in higher plant. So first question, describe Calvin cycle with schematic representation. These are the points, you can read it. Here I given Calvin cycle schematic representation, you can refer it. And here main three steps are there. First one, carboxylation. Second one, reduction. Third one, regeneration. And next question, state Blackman law of limiting factor and explain the 4 external factors affecting photosynthesis. So here statement is there, and so 4 factors are there, it is affecting for the photosynthesis. Next question is there, what is Cranch anatomy? Explain the Hatch-Slack cycle with the help of diagrammatic representation. Here points are there. And last I given schematic representation. Next question is there, list out the difference between cyclic and non-cyclic photophosphorylation. Second one is there, PS1 and PS2. Third one is there, C3 and C4 cycle. So first difference we will study, that is C3 plant and C4 plant. C3 means that is Calvin cycle pathway. C4 means Hatch-Slack pathway. C3 it contain 3 carbon compounds, C4 it contain 4 carbon compound. And C3 no Cranch anatomy, so in C4 presence of Cranch anatomy. And in C3 CO2 fixation enzyme, that is Rubisco is there, so in C4 CO2 fixation enzyme is PEP carboxylase. And in C3 optimum temperature is there 15 to 25. In C4 optimum temperature is at 30 to 40 degrees Celsius. And C3 example wheat, rice, barley. And C4 example maize, sugarcane, sorghum. And next difference between that is PS1 and PS2 is there. PS1 absorbs the light at 700 nanometer. PS2 absorbs light at a 680 nanometer. Next point, location in the chloroplast found in the stroma, thylakoid and non-oppressed region. Location in the chloroplast found in the grana thylakoids, that is oppressed region is there in photosystem 2. And next is there, it reduce NADPH. And next point is there, that is photosystem 1, location in the chloroplast found in the stroma, thylakoid and non-oppressed region. And in photosystem 2, location in chloroplast found in the grana thylakoid, that is oppressed region. And next point, it produce NADPH during light reaction, and it split water molecules to release oxygen. And in photosystem 1 do not evolve oxygen, but here they release the oxygen by photolysis of water. And in PS1 electron acceptor ferredoxin is there, and in PS2 electron acceptor is plastoquinone is there. And next difference between cyclic and non-cyclic photophosphorylation is there. These are the points. That is, first point is there, electron follow a cyclic path and return to the same PS1. And in non-cyclic, electron flow a linear pathway, do not return into the original photosystem, and it contain only photosystem 1, but here they contain photosystem 1 and photosystem 2. And in cyclic photophosphorylation, no oxygen release. In non-cyclic, oxygen is released. In cyclic photophosphorylation, no NADPH form, and in non-cyclic NADPH form. And in cyclic photophosphorylation, they produce only ATP, but here they produce ATP plus NADPH. Chapter is there, chapter number 12, that is respiration in plant. So the first question is there, give the schematic representation of Krebs cycle, or it is also called as TCA cycle, or it is also called as citric acid cycle. You have to practice this cycle. And next question is there, glycolysis is often referred to as a EMP pathway. Why? Give the schematic representation of glycolysis. Here they are asking question, why glycolysis is also called as EMP pathway? Because it is discovered by three scientists, that is Abenton, Meherov and Parnas. So that scientist initial letters they taken and they make one pathway, that is EMP pathway is there. And the next schematic representation of glycolysis, half schematic representation of glycolysis is there. Here you can refer it and you have to practice this schematic representation. Next question is there, what is RQ value? Mention RQ value of the fat, protein and carbohydrate. RQ value definition is the volume of the CO2 released over the volume of oxygen absorbed during respiration. So that RQ value of fat is 0.7, carbohydrate is 1, protein 0.9. And what is fermentation? So fermentation means a biochemical process that breakdown of the glucose to produce the energy and other substance like lactic acid or ethanol in the absence of the oxygen, means partial breakdown is takes place. So that's why they're releasing little amount of energy and plus some other chemicals. So that's why we are calling as a fermentation. And next is a schematic representation of ETC, electron transport chain. Remaining chapters I will cover in part 3 video.